In this video, I'm going to talk about genome organization, or what's in a genome, what makes up the genome. This is part one of a few videos. Chromosomes contain all or at least some of the genomic DNA of an organism. When I said the word chromosome, probably something like this X shape appeared in your mind. This X shape is a mitotic chromosome. Mitotic meaning this is from a cell that is undergoing mitosis. It's in the process of cell division. So what you're actually looking at here are two sister chromatids. We call these sister chromatids, even though this is one chromosome, because each arm of this chromosome, this little sister chromatid, is the product of cell division. And what that means is that these two sister chromatids are identical in genetic sequence to one another, barring any spontaneous mutations that might have occurred during cell division. And here we see two mitotic chromosomes. On the previous slide, that was one mitotic chromosome with two sister chromatids. Here we're showing homologous chromosomes. So these are still mitotic. You are still seeing these sister chromatids of each of these chromosomes. But here we have homologous chromosomes. So for a diploid individual, you might think that one of these chromosomes came from the maternal parent and one came from the paternal parent. And an organism can have more than one chromosome. Its genome can be spread out across multiple chromosomes. So here, what I've diagrammed are three pairs of non-homologous mitotic chromosomes. So we still have the homologous chromosomes, like shown here. This is, let's call this chromosome 1, the maternal copy, chromosome 2, the paternal copy. And each of these chromosomes is composed of two sister chromatids because of the particular stage of mitosis that we're looking at right now with this individual. And you can see I've drawn, we can call this chromosome 2 and chromosome 3. These chromosomes, these non-homologous chromosomes, they appear differently. And we usually number chromosomes by size. So chromosome 1 might be the biggest, and chromosome 2, you can see it's a little bit shorter. Other ways that chromosomes can be different from one another, non-homologous chromosomes, is that this middle bit, the centromere, might be in a different position. So you can see here, these centromeres on chromosomes 1 and 2 are pretty much in the middle of the chromosome, but they can be nearer the end. They can be acrocentric. So again, what I've drawn here are three non-homologous chromosomes. So what you might say is that this organism that I've drawn here has a haploid genome size of n equals 3, and it has a diploid genome size of 2n equals 6. If you zoom in on any region of a mitotic chromosome, you're going to see that there is compact chromatin. Chromatin is DNA packaged tightly with lots of proteins. There's some form of high order compaction that is compacting that chromatin to make up the mitotic chromosome. Now, if you zoom in on any piece of compact chromatin, you'll start to see what we call the 30 nanometer fiber. The 30 nanometer fiber, now for the first time, you are seeing our nice double-stranded DNA in this teal color, and it's bound up in proteins, a collection of proteins called the nucleosome, which is gold in this form, the solenoid form of the 30 nanometer fiber, whereby these nucleosomes arrange themselves in this circular pattern, or another alternative version of the 30 nanometer fiber, the zigzag form, where nucleosomes, which are also coded purple here, so you can see the zigzagging formation along the 30 nanometer fiber. The details of the orientation here are not that important, but one thing that I would like you to notice is that differences in how the DNA is packaged with proteins can affect which sections of DNA are physically close to one another. So, for example, I'm going to put a little star on this section of DNA. That's a DNA sequence. And in the solenoid form, that section of DNA, that DNA sequence, is physically close to that yellow star on nucleosome 2. Now, if I starred that same sequence of DNA that's associated with nucleosome 1 over here in the zigzag formation, and then I star that same DNA sequence that's associated with nucleosome 2 in the zigzag formation, you'll see that what was physically close in the solenoid formation, those DNA sequences between nucleosome 1 and nucleosome 2, are now farther away. So depending on how DNA is packaged can affect how physically close and 3D physical space DNA sequences are to one another. And now if you were to stretch out that 30 nanometer fiber, you would see something like this beads on a string formation that I have kind of sloppily sketched for you here. Here's a much better representation of the beads on the string. So the beads part of the beads on the string are nucleosomes, and these nucleosomes are made of histone octamers, that is 8 histone proteins. Histones are proteins that associate with DNA, and there are 8 of them in each one of those beads or the histone octamers. Wrapped around that histone octamer that you can see here with this DNA is about 150 base pairs of DNA that is associating with that protein. You'll also see that there is another histone protein up here, H1, which is not part of the core histone, but it is associated with the DNA as well, and it's responsible for helping form some of those higher order compaction structures like we saw in the previous slide with the solenoid and zigzag. And that H1 protein is also associated, as you can see here, with this linker DNA, which is about 20 to 80 base pairs of DNA, depending on the formation of the proteins. And now finally, after all that compaction to get to the mitotic chromosome, the compact chromatin 30 nanometer fiber beads on a string, we have, of course, wrapped around these beads is the string, our favorite string, naked DNA, shown here, sloppily drawn in its double helical form of 2 strands bound together with interactions between the bases. Okay, but what's in the DNA? This video is getting a little bit long, so let's answer that question in part 2.